Hi everyone, my name is Kara Perkerson. Today I'm going to be talking to you about my project. It is entitled Investigating the Interactions Between NTHI Surface Proteins, OMP26, and Protein D. Before I dive in, I'm just going to give you an outline of what I'll be talking about today. I'm going to give you an introduction to acute otitis media, and then an introduction to vaccine development, how it is developed, and the challenges with them. Then I'll give you a background on OMP26 and protein D. From there, I'll talk about the studies that I performed during my time here at RIT, the conclusions that we generated from there, and future work we plan to do. So acute otitis media is also known as a middle ear infection. I'm sure most of you have had this in your lifetime, at least once or twice. This occurs in the middle ear space of your ear. So this outer part here is the ear canal. Then you have an eardrum, which is just a clear membrane, and then behind the eardrum is your middle ear space. This also contains your ear bones, and then from there, that goes into your balance organs and your hearing organs. It's a very critical space where everything that comprises the ear connects. And from there is the eustachian tube that goes to your nose. So your nose to your ears all connected. When you get an ear infection, you'll get a slew of symptoms. This can include a fever, common with a lot of infections, temporary hearing loss, you can get some ear pain, ear irritability, decreased appetite, and then even fluid in your ear. So our normal eardrum looks like this. As you can see, the eardrum is clear, the skin isn't irritated at all, it's not red, there's no fluid, so it's a nice healthy eardrum. But when it's infected, as you can see, it's red, there's fluid pushing against the eardrum, it obviously wants to come out, it's bulging, so this is going to cause you a lot of pain, a lot of irritability, and with all that fluid, it can also ensure that you're not going to be able to hear, and that's quite a problem. So with ear infections, they're really, really common in young children, and they tend to reoccur in young children. So the question is, why are children so prone to ear infections? So physiologically, your eustachian tube, as an adult, your eustachian tube is quite long and it has quite a steep curve before it gets to your middle ear. So it's a really long tube. It'll go up a little bit and then it goes to your middle ear. The whole purpose of your middle ear space is to kind of drain fluids generated by your eardrum and anything else in that space. It actually comes through your eustachian tube to your nose. But with infants, when they're born, when they're really young, that eustachian tube is a lot shorter. And also with it being a lot shorter, it's a lot more shallower. It doesn't have as steep a curve as an adult has. And so when fluids drain out, it's a lot quicker than an adult. But when you get sick, say with the cold, and you start sniffling, and it gets clogged, so you start pushing back the fluid that's trying to be drained out back into the middle ear space. In adults, because it's so much longer and steeper, it's not going to be as easy to do that, and therefore you're not going to get an ear infection after you've been sick. But with infants, just a minor cold can push that fluid back really easily into that middle ear space, and they're not easily get an ear infection. But then the other reason they can get recurrent ear infection is their immune system. When they're young, when they're born, they have a really naive immune system, meaning that they haven't been exposed to enough microbial germs and enough bacteria to have a nice, strong, healthy defense system against bacteria. And because of this, they're going to be a lot more easily prone to infections, to illnesses. This is why when you're seeing a baby, you need to make sure that you yourselves are healthy so that way they can stay healthy. However, when they're about 5 to 6 years old, their immune system is a lot more developed and it has better defense systems, and they are a lot more healthy because they have been exposed to a lot more bacteria and germs now. And with that, you see a very large decrease in the number of recurrent infections in children of that age, to possibly no more ear infections at all in their life. Some people do have recurring infections throughout their life, myself included, but that's probably just a genetic component or they're just more prone to it. But for a large amount of people, by the time they hit about 6 years old, they could possibly never have another ear infection again. So in the US, because so many kids get recurring ear infections, it is the most diagnosed childhood illness in the US. And with that, it is responsible for more than 30 million clinic visits annually. Now, this creates a huge burden on the healthcare system, on insurance, and on us as people. 30 million clinic visits doesn't represent that being one child each. It could be a child going for multiple visits, or siblings, or adult. However, that's a large amount of doctor visits. And with ear infections, they tend to go away on their own unless they're severe. But a lot of parents don't know this, a lot of people don't know this, and therefore they want treatment for it. They think it needs to be treated somehow. So it is also the most common infection in which antibiotics are prescribed to children. And the reason it has to be antibiotics is these are bacterial infections and not viral. So you can't just get some sort of over-the-counter drug. It has to be antibiotics against it. The issue with that is the emergence of antibiotic-resistant bacteria. And for children to get antibiotics at such a young age, there's been an issue with overprescription of antibiotics. And with that, that could later create another burden on the healthcare system of they are now resistant to these antibiotics and therefore need treatment in the hospital with IV antibiotics or some case like that. However, with acute otitis media, it causes an economic burden of more than $5 billion a year. That is an extraordinary amount of money that has been placed on the healthcare system and on the insurance system. And there is a major need for this to be decreased in the prevention of ear infections rather than the treatment of ear infections, because there are multiple different types of bacteria that cause ear infections. So these bacteria are listed here. Streptococcus pneumoniae, otherwise known as S. pneumoniae, is a respiratory bacteria. It primarily causes pneumonia. However, it is pretty consistent with the common cold and also with ear infections. It accounts for over a third of all cases, 35 to 40% of all cases. This is not going to be the focus of our talk today, as there is already a treatment option available for this specific bacteria. Non-typable Haemophilus influenza is next. It accounts for about 20% to 30% of all cases. Non-typable Haemophilus influenza, instead of just Haemophilus influenza, is critical because non-typable means that instead of a uniform outer layer, it actually doesn't have that. It's missing that. Therefore, the surface is comprised of thousands of different surface proteins and receptors, etc. There is treatment against the uniform bilayer Haemophilus influenza. However, there is not for non-typable Haemophilus influenza. And then the last one is Varicella catarrhalis. This accounts for 10% of all cases. Because it only accounts for 10%, it is also not necessarily needed for prevention yet. Right now, we're going to be focusing on NTHI, non-typable Haemophilus influenza. So current treatment of acute otitis media, as said before, are antibiotics. They are really commonly prescribed to children, typically amoxicillin, as it's much gentler on their immune systems in their bodies. Problem is the emergence of antibiotic-resistant bacteria, and this is because we have been overprescribing antibiotics. In reality, what doctors should be practicing is if a child has an ear infection, they come into the clinic for a visit. If they haven't had it for more than 3 days, they need to be sent home without any treatment. If they are sent home with treatment, when an ear infection actually goes away on its own, then that just kind of encourages this rise of antibiotic-resistant bacteria, which you really don't want to be exposed to later in life. It can be really harmful to your body. So doctors are trying to just make sure that they're not treated unless it is a severe ear infection, in which antibiotics are the only means necessary for them to treat children. And then there's prevention. And this prevention comes in the form of a pneumococcal conjugate vaccine. This protects against only some of the most violent strains of pneumonia, not all. And that's because there's a difficulty in generating a vaccine in which you can have broad coverage. It might not contain the surface component that you want in the other strains that you have in this vaccine that it protects against. However, because this protects against S. pneumoniae, and specifically the strains that you want to be protected in, we've all had this vaccine, and therefore it is not a concern for us as much as NTHI is a concern. So there are multiple different types of vaccine. There are three main types. There's an inactivated vaccine. An inactivated vaccine is essentially a killed bacteria or a virus. So it has all the components it contains as if it was live, but it is not live. It will not be actively replicating. It will not be performing the same methods as does. It just contains all of the components as if it was. And that will be then injected into the host for the immune system to build up some immunity against. Some examples are your yearly flu vaccine. The one that goes in your arm, that's an inactivated vaccine. Another one is polio or hepatitis A or even rabies. Those are all inactivated vaccines that we commonly see. The next version is a live vaccine. This is an attenuated bacteria or virus. An attenuated means that it's a weakened version rather than the full pathogenic bacteria or virus. And it tends to be more temperature sensitive. It tends to only replicate in certain parts of the body, especially those where the pathogenic version will reside. And this is really beneficial because it is a true defense for the immune system to recognize and generate antibodies against, as it is a version of the pathogenic bacteria. However, this is a problem for children under the age of 2. They still have a naive immune system, meaning they can get quite sick from a live vaccine, as well as those that are immunocompromised and have autoimmune diseases cannot get these vaccines, which is why we are so reliant on herd immunity, especially with live vaccines. We have to make sure that a certain number of people have had the vaccines to protect those that physically cannot get them. So some examples are the chickenpox vaccine, the MMR vaccine, shingles, smallpox, etc. Immunocompromised people cannot get these vaccines. And as we know, they're really contagious. So we have to make sure that we are protecting those people. We don't really want to do a live vaccine if we can do an inactivated or a subunit vaccine and benefit everyone, especially since we're focusing on kids. A live vaccine will not benefit us here. So the last type is a subunit. And subunits can come in multiple parts. There's a protein-based subunit vaccine, meaning that we actually pick a surface protein and then we make more of those and we put it in the host, and the immune system will generate antibodies just against those proteins. So when the pathogenic bacteria comes in, it will see the protein and it will go, oh, I have antibodies for those, and generate them. Same thing with toxoid. It uses a toxin instead of a protein unit. So a really great example of a toxoid is the DTaP vaccine. We all have it. We all get it more than once. That is the only widely marketed toxoid vaccine as known. And then there's a whole pathogen subunit. A whole pathogen subunit is similar to an inactivated, but instead of having all the components, it is just a shell that is put into the body. There's nothing else in it. It is a pure shell. So some examples of the subunits are Hep B, the Hib, some influenza vaccines are subunits, the pneumococcal vaccine, the HPV, et cetera. What's been great with vaccine development is there has been a large increase in bacterial vaccines rather than just viral vaccines. So this is a really promising developing field for in the future when we're not going to have to be so reliant on antibiotics when we can just prevent the full force of pathogenic bacteria on our body. So here we're going to focus on a protein-based vaccine. Just going to give you a little clip through. So when you get vaccinated, you're going to have this protein emitted from it. It's going to go into your body, and then the immune system's going to see it and make antibodies against it. And it'll make a bunch of them, and some of them will be stored as memory. So what makes a good vaccine candidate? It needs to be really highly specific for the target, for the bacteria itself. So it should only be able to, for the immune system to look at the bacteria, see the target, and go, oh, I need to make antibodies against it and stop it from replicating. It needs to also be highly conserved among strains. A lot of bacteria and viruses have hundreds, maybe thousands of different strains. So we need to find a candidate that is preserved among all of them, that is present amongst all of them. You can typically see this with sequencing, but picking a candidate that's there in all of them means that there's a really high broad protection, really high broad coverage, and therefore you will be protected against everything, and you're not going to have to worry that you're not protected against certain strains. So say the influenza vaccine will only protect against certain strains a year, but if you get a strain that you're not protected against, then it's like you never had a vaccine. It also needs to be surface exposed. This is critical. This is the first line of defense in the immune systems, that they're going to see something on the surface that doesn't match with what's typically regulated in the body, and it needs to be able to find an antibody to inhibit it. It also needs to be antigenic. It needs to make a strong antibody response. So because we put a small amount in our body, it's not like we're actually sick, we're just mimicking it. And the immune system needs to see that and be able to generate enough antibodies that it actually retains a memory from it. And from there, when you actually get sick, because it retained the memory in it, because it retains some antibodies against it, it will be able to generate an even higher antibody response the next time. So in collaboration with RGH, we've been doing this project. They have identified 4 really strong vaccine candidates against NTHI. These include OMP26, Protein D, P6, and Protein F. All of these have been published as vaccine candidates and even in vaccines themselves. They are found to be pretty conserved among a lot of the strains of NTHI, and they have some really promising preliminary data that has narrowed it down to these 4 candidates for NTHI vaccine. But today for this talk, we're just going to focus on OMP26 and Protein D. Just wanted to expose you to the other 2 as you're just going to see it's present in the preliminary data. So to give some background on the proteins themselves, OMP26 is also known as outer membrane protein 26. It is 26 kilodaltons in size, so it's a fairly small protein. However, it is conserved among all of the NTHI strains. So that's good. It has broad coverage, just like we want as part of it. It is similar to skip protein, so skip is its homolog, as we have found in E. coli. And skip has a role as a molecular chaperone. So we think OMP26 may also have a role as a molecular chaperone, and that its shape is like a donut, essentially, with its active site in the middle. It is not confirmed, it's not been characterized, and its structure has never been experimentally confirmed either. But the group at RGH had done some preliminary studies where they had infants in the range of age from 6 to 30 months old. So they're almost 3 years old at this point. And they all had ear infections. So what they did was they took samples from their middle ear space uh, while they were having the ear infection to test for the amount of antibodies that were generated against some of the vaccine candidates um, from their body. And so in this study, they found that OMP26 had a high number of antibodies generated against it. They had 500 nanograms per mil amount of antibodies generated against it, which is quite a high number. So this tells us that it's immunogenic and it could be antigenic as well. So that checks off three large boxes. This has broad coverage, it's on the surface, and it's an antigenic protein, and it's already been published before as a vaccine candidate. So OMP26 became a very strong candidate. Protein D is a little bit larger than OMP26. It's once again a surface protein. It's on the outer membrane. It is 42 kilodaltons, and it is also conservative on all the MTHI strains. So once again, broad coverage and surface. It's already checked into the boxes. It has a proposed function to adhering to epithelial cells. Epithelial cells line your skin and your muscle. So therefore it has to be a surface. So it has to stick to something. In the same study as the OMP26 protein, they also looked at antibodies generated against protein D. And this generated a really high number of antibodies against it, higher than OMP26. And this tells us that it's really immunogenic and it's really antigenic in infants. Also, another major benefit of this protein is that it's already part of the pneumococcal conjugate vaccine. So it's already playing a role in a really important vaccine that we all get as children. And therefore, this is a candidate that has to be put into our anti-HIV vaccine. So with these candidates, with the 4 candidates, they did preliminary studies using a mouse model so that we could see the amount of antibodies that were generated, as well as bacteria generated when they have had a vaccine versus without. So mice that had the vaccine were given 3 doses to ensure the best amount of antibodies generated as possible. So 5 weeks before they were infected, they got their first vaccine. A week later, they got their second dosage. And then 2 weeks after that, they got their last dosage. Those mice were then left for about a week or 2. And after 2 weeks, they had some blood samples removed. So that way we could test the amount of antibodies that were present in their body against these vaccine candidates. After it had been bled, we gave them the PR8 flu virus intranasally. The reason we did that was to prime mice to mimic an ear infection. Mice cannot naturally get ear infections, so we have to prime them as if they're going to get a respiratory bacterial infection. So intranasally, because you get a lot of your respiratory infections in the nose, we gave them the flu virus to prime them. And then a week later, we challenged it with the bacteria that we want to study. So we challenged them with NTHI, also again intranasally. And then 2 to 3 days later, we harvested the mice, and then we washed their nose and took samples. We washed their ears and took samples, and then we also took their ebrula to look at how much bacteria was left in their body as they were harvested. And it had to be 2 to 3 days, because if we waited too long, they would clear out the bacteria completely, or they would die if we had given them too much. So 2 to 3 days was a prime time for harvesting. To make it simpler in words, we vaccinated the mice with the vaccine candidates, and these vaccine candidates all included an adjuvant. Adjuvants are common in vaccines, and they help the vaccines continue to be stable for a longer period of time. So we found that antibodies peaked 2 weeks after the last booster, meaning that we wanted to take the blood samples 2 weeks after we gave them that third dosage. We then infected them with the flu virus and then the bacteria and harvested them once again. So these were the vaccine formulations that we decided to study. We had to do a control for the adjuvant, so we used aluminum. This is a very common adjuvant in vaccines. Then we tried 3 different versions of formulations. We had a trivalent vaccine. This contained protein D, OMP26, and P6. Then we had 3 divalent versions. We had OMP26 and protein D, P6 and protein D, and then P6 and OMP26. Then we had 2 monovalents, the P6 only and then protein D only. We did not test OMP26 only, as we knew that it wouldn't generate a strong enough antibody response itself. So here we have some preliminary data. All of these data we're looking at the amount of antibodies generated against protein D itself. So we took all those vaccines, put them in mice, infected them, and harvested them. But we were wanting to look at what antibodies were generated against just protein D. We don't really care so much about the other ones. What we found was the aluminum control did not have an effect, which was good. The trivalent vaccines, this contained OMP26, protein D, and P6, had such a low number of antibodies generated against it, against protein D. And this is really interesting because it's just such a low number. Same thing with OMP26 and protein D divalent vaccine. There's almost no antibodies generated against protein D in either of these cases. But when you do the protein D and P6 version, there's a large number of antibodies generated against protein D. And then protein D alone has a really high number of antibodies generated against it. So why is it that when it's mixed with P6, it has this high number of antibodies generated against it, but when it's mixed with OMP26, it doesn't? And when it's mixed with OMP26 and P6, there's no antibodies generated against protein D. And this started to get us thinking, what is happening here? There's got to be something happening. So this was rerun. Once again, we looked at antibodies only against protein D. And when you have protein D alone, so you can see there's a large number of antibodies generated against it. But with the trivalent, there's a low number of antibodies made against protein D. We then tried the trivalent vaccine in separate shots. So we gave all 3 proteins separately. So we gave 1 protein in one leg, 1 protein in another leg, and then 1 protein higher up to see if that had an effect in the lymph node generation of antibodies. And it only generated maybe a medium antibody count, but it's not as high as protein D by itself. OMP26 and protein D mixed together once again, there's no antibodies generated against protein D. But when we gave it separately, so we gave protein D in one leg on the right side, say, and then OMP26 on the other leg. This should have generated a high number of antibodies against protein D. It should have been very similar to as if protein D is alone, because you have lymph nodes in your joint. That's where you make antibodies. However, only generate a medium antibody count once again. So we're starting to think somehow it's finding each other in the body and interacting and preventing antibodies being made against protein D. So it starts making it, and then it's somehow then inhibited to where it's not. So the hypothesis is that OMP26 is either binding to or interacting with protein D, and this is preventing and inhibiting the protein D antibody response. So the goal of our work is to investigate how and why protein D and OMP26 are interacting in vivo. to reduce that antibody response against protein D. So our approach is going to be to utilize the in vitro version, so in vitro interaction and binding assays, to basically confirm and quantify their binding and their interactions together. So before we can do that, we actually had to express and purify the proteins themselves. So we would streak a plate with a plate. This contained LB. LB is a media that E. coli likes to feed on and grow on. E. coli would contain the gene that would express the protein we wanted. So it would express the protein of interest. So we'd have one that would express protein D and one that would express AMP26. We would then grow it in liquid media, and this would overexpress that protein production in E. coli. So we would want E. coli to make as much of that protein as possible. We'll then harvest those cells. There shouldn't be anything in the supernatant. It's all junk, so that was removed and cell pellet was stored. When we were ready for purification, we would resuspend the cell pellet in the correct lysis buffer and sonicate them to lyse the cells. So we would break open the cells and extract the protein that we were interested in. So once those cells were broken up, we would spin them down at a high rate of speed, in which the broken cells consist of the pellet and all of our protein of interest is in the supernatant. So the pellet was discarded, the supernatant was saved, and we would purify using talon beads. Talon beads are a metal affinity bead. So most proteins, a lot of proteins in biochemistry, are genetically modified to contain an N-terminal cis-his tag. This cis-his tag has a really high affinity for these talon beads. So the benefit with these is that when we load the supernatant on the column, because they have this tag, as they go through the beads, they should bind onto the beads. And therefore, any junk proteins that don't have this tag and proteins we are not interested in will flow right through the column. They shouldn't bind to anything. And then once we've ensured that all of the junk is out, we would elute the protein from the beads by breaking that bond using imidazole and come out. And this way we can ensure that we have a solution consisting of just one protein. So in this image, these are protein D and OMP26 separately, as well as a premixed version of protein D and OMP26 I've done overnight after purification. All of these proteins were exchanged into a salt buffer for storage. This lysis buffer is not so great for long-term storage. I also had to dilute protein D as we had such a high concentration of it generated from the overexpression. We had about 22 mg per ml, whereas OMP26 had about 1, which is great because we had a ton of protein we could work with. So as you can see, in the protein D lanes, there's protein D at 42 kilodaltons, right the size we want. And it is also present in the MITs at that correct band. And then with OMP26, it's at 26 kilodaltons as well as in the MITs. So as in the MITs, as you can see, there are 2 clear bands. Those are the 2 protein presents, and therefore they're pure. Same with protein D. With OMP26, there's a little bit of cross-reactivity. We think that somehow a little bit of protein D is also expressed and purified with OMP26 as part of the genetic component. However, OMP26 is fairly pure. We wanted to further confirm it with a Western blot. A Western blot is where we can attach an antibody that's highly specific against the protein of interest, and then a secondary antibody on top of that with a fluorescence tag. So that way we can fluorescence see there should just be one band for that one protein. So with the OMP26 blot, this was used with anti-OMP26. In the MITs, you can clearly see OMP26 at 26 kilodaltons, as well as in the OMP26 lane, no protein D in the protein D lane. There is some cross-reactivity with protein D, but as we have mentioned, we think it also gets expressed in the AMP26 gene. However, we can confirm that AMP26 is present in the correct samples. And then protein D, once again, we can see that there's protein D at 42 kilodaltons in its own lanes and in the MITs. And there's that little bit of cross-reactivity with AMP26 that was also seen in the AMP26 blot. But it confirmed that protein D is present. To further confirm that it was pure, we did size exclusion chromatography with Dr. Glughorn. So we ran a size exclusion column on our FPLC. The blue peak is protein D only. As you can see, there's one very nice sharp peak, which is where protein D was eluted. And then the yellow peak was AMP26, where AMP26 was eluted. And then the green peak was where AMP26 and then protein D were eluted. So there is one clear peak per protein, and then the one containing the MITs had matching peaks to the correct protein. And Mangels confirmed that they were very pure and nothing else was contaminated. So this confirmed our pure proteins. So from there, we went to a co-immunoprecipitation study. Co-immunoprecipitation is a binding assay typically used in immunology to look at receptors and antigens and proteins, etc. It is an older method, but I thought it was a method that was needed to be done to look at our binding interactions. So what we had done for this specific blot, there are 4 different types of results, was we had taken protein D and AMP26 and mixed them together. I mixed them together spinning overnight to make sure they were homogeneous. They were 50-50 protein D and AMP26. There's no differing ratios. We just wanted to confirm that the experiment worked. At the same time, I had incubated magnetic beads with a specific antibody. So for this one, I'd incubated it with anti-protein D. This way, anti-protein D is on the surface of the beads, and hypothetically, because protein D and AMP26 are binding, protein D should bind to anti-protein D, and it should also bring AMP26 along with it. So anything that is bound to the magnetic beads will be considered a bound, and anything that was not bound is unbound. So we ran an SDS page shell and a Western blot after this experiment was performed. There was no protein D found in, or AMP26 found in the unbound. So this blot was detected with anti-AMP26. So we're looking for AMP26 rather than protein D, even though the magnetic beads were conjugated with protein D. So in the bound sample of protein D, we can see that AMP26 is present. So that means that it was binding to protein D, which bound to anti-protein D. So AMP26 is present, meaning that it's binding to protein D somehow. And there's no control, there's no band present in the protein D control, as there is no AMP26 present. And we are at 26 kilodaltons. Because there's no AMP26 present, that's blank. But there is AMP26 present in the bound sample. So this is telling me that they are interacting. So once again, this blot was detected using AMP26, but this time the beads were conjugated with anti-AMP26. So this means that if they're binding, it will be AMP26 and protein D bound together, and then AMP26 will also be bound to the antibody. So no AMP26 was found to have flown through. That means that they all bound to a bead, or to an antibody specifically. And there was AMP26 confirmed to be bound to the magnetic bead as well as the control. So this confirmed that the experiment worked really well. It was there. So now we're detecting the protein D version. So we're looking at anti-protein D. So we're back to the first original experiment where we had seen that OMP26 is present when it's bound to beads with anti-protein D. So this means that protein D should also be present as well. It wouldn't be present otherwise. And as you can see in the control and the bound protein D sample, they are present. And there's no unbound protein D that was found to flow through. So all of them bound to some anti-protein D. They're there. So that means that they have been interacting, they were able to bind. For this one, we conjugated the magnetic beads with anti-OMP26. So there, and we've seen that OMP26 is bound to the anti-OMP26. So now we're detecting the blot with protein D. So theoretically, if OMP26 was bound to the beads, protein D should also be bound to the beads. And we can see that very clearly here. We can see in the bound sample, protein D is present. There's also a little bit present in the control, but that is due to the cross-reactivity. And there is none present in the unbound samples. So this confirmed that they are interacting once again. So just a summary, we took purified OMP26 and protein D recombinant proteins. They were pre-mixed overnight in a solution. With anti-OMP26 conjugated beads, we were able to pull out OMP26 and protein D. This was the same case for anti-protein D conjugated beads. So this suggests that OMP26 and protein D proteins do bind and do interact in vitro. So the next step was to do an ELISA. And we wanted to probe this with anti-OMP26. So an ELISA is an enzyme-linked immunosorbent assay. So this is another binding assay commonly used in immunology. What we wanted to do here is we wanted to coat the plate with the protein of interest, and then from there we should be able to see some binding between proteins and between the antibodies. And the issue we were having was when we would coat it with protein D, for some reason, OMP26 would not stick. But if we coated it with OMP26, protein D would stick. And there were issues being had with this experiment. So in conclusion, we were unable to detect that there was binding between OMP26 and protein D, and if there was a complex that was occurring there. So this had us starting to think that there must be a different way we have to look at our binding assays, as a typical ELISA was actually not representative of what we were trying to look at. We started thinking that maybe it wasn't just binding, that they were just somehow interacting with each other instead of fully binding together. So in conclusion, we were able to successfully express and purify recombinant non-lipidated OMP26 and protein D. We confirmed their purity by using size-dissolution chromatography. The co-immunoprecipitation studies support the hypothesis that they do interact in vitro. However, the ELISA studies were inconclusive, and we were unable to detect these binding between OMP26 and protein D. So we have initiated crystallization studies with the Glughorn lab to investigate their structures. So we want to investigate protein D and OMP26 structures separately, as well as a possible complex that they make together. So preliminary results with these studies suggest that their interaction is actually quite sensitive to the amount of salt that's in the buffer, and the concentration of the buffer, and the pH of the buffer. So this leads to our future work. We want to try co-immunoprecipitation, as it has confirmed to work, with different ratios of proteins. So we had done the initial studies with 50-50 of each protein. So we want to try maybe 80-20 or 90-10, etc. We also want to try them in other experiments with the differing buffer conditions all into immunoprecipitations. We also want to try a fission experiment. So we will utilize histad protein and talon bead columns in which, say, we have histad 26, but then we have protein D that doesn't have a histad. Let them mix and see if they bind and see if it's strong enough for it to not even elute in the flow through, where it elutes with the histad 26 itself. So those are some experiments we want to do. We also want to further the crystallization studies with the Bloodhorn lab and get X-ray crystallography of OMP26 by itself, protein D by itself, and then the complex. And hopefully the studies of these structures will help us look at, okay, we know the structures and we know where the active site is, so how are they interacting? Are they interacting in the bindings in the active site and binding, or are they just interacting covalently? So these are really critical experiments that we want to focus on in the near future. So just to finish off, I would like to thank Dr. Michelle for the last 6 years in this lab. It's been a great experience, and I'm definitely bittersweet to leave. I also want to thank Dr. Ravinder Kaur from RGH, Dr. Bloodhorn from RIT, helping us with all the crystallization and size exclusion studies, and then Dr. Patricio from RGH for funding all of these studies. I also want to thank the Rochester Bridges Doctorate Program for their support with me throughout this master's. These are all of my directors and coordinators, and they've been a huge help in supporting me through these last couple of years. And then within the Michelle Lab, I specifically want to give a thanks to Naya, Susan, and Wilford. They have been my little team supporting me throughout all of these experiments and all of the craziness. And I just really want to express a heartfelt thanks to all of you for watching, to all of you in the chemistry department for supporting me, and I will miss you all. Thank you so much.